anatomists and physiologists. This video is another video in our blood vessel series. This is about capillary exchange. When blood moves through our capillaries, it goes really slowly. And what I mean by slowly is really slowly. And this is really important because at the capillary level, we need lots of exchange to happen. We need to get rid of put oxygen into our tissues, put carbon dioxide back in that blood so we can bring it back to the lungs, and then nutrients and all of the other things that we care about. So we need time for all of that to happen. So it's good that capillaries are slow. And how a lot of substances get across that capillary from the inside of that capillary to the outside of the capillary into the tissue is mostly diffusion, which is nice because we already know about that word. Diffusion is, if we remember, it's when the substance travels from a high concentration to a low concentration. So for example, in our respiratory gases, for example, oxygen and carbon dioxide, I'll actually write that on our slides here. Respiratory gases like oxygen, like CO2, carbon dioxide. Nutrients, metabolic waste, all of this stuff can go by diffusion, and there's a couple of different flavors of diffusion that we've talked about before. So if something is going from a high concentration to a low concentration, that would travel across the membrane in whatever way, or across that capillary wall, okay? So some common things that we'll need to remember is that when we're inside, so if I'm drawing a capillary, and this is the inside of my capillary, and this is the fluid around, like this is all that fluid around. We would call the inside of that capillary, we call that the lumen, L-U-M-E-N. That's the inside part. That's where all the blood cells are, and the liquid, and all the other things. On the outside of the capillary is what's called the interstitial space. Interstitial space, or the ISS, interstitial space, and that's usually fluid. So oftentimes you'll hear that called interstitial fluid. So just be aware of that too. And then of course on the side would be ISS as well. Interstitial space. Again, usually full of fluid and junk. Okay, and this is where our tissues are. So we travel through all of that interstitial fluid, and then we'd have our tissue cells. And these are the tissue cells. Whatever tissue that we're in. We'll put like cute little nuclei in there. Okay, so how exchange then will happen is that we want to leave the blood vessel or enter the blood vessel, travel through that interstitial fluid or interstitial space, and then to our tissue cells. And we can do this, your textbook says, by diffusion in 4 ways, and it kind of depends on what kind of substance that we are. So I'll highlight all of these as I'm talking about them. So step 1, or option 1, I suppose, is diffusion still. It's for lipid-soluble substances. So something that's fatty or something that's soluble in fat is a lipid. So lipid-soluble substances, fat-soluble stuff, can just pass through. That cell, because lipid-soluble things can also pass through the plasma membrane. Okay. The next thing is for our substances that are water-soluble. So water-soluble substances are not lipid-soluble, so they can't just pass through that membrane. They need another method of transport, and that method of transport is through the intercellular cleft. That's that little gap in the cells, that little space. So they just travel through space if they're water-soluble. The next option is also for water-soluble things. So water-soluble things can go through these little intercellular clefts, or we can pass through fenestrations. It's the little pores in fenestrated capillaries, little pores. And then if you're a really big substance, then you can transport by vesicles. So these vesicles are when our little substance would be engulfed by the membrane, transported across the membrane, and then exocytosis would occur on the other side. And then of course this can go either direction, so it can go this way or it can go that way. So we engulf it, that vesicle travels across, and then we release it. So capillaries can do diffusion because the stuff will travel from a high concentration to a low concentration, whichever direction that is. Okay. But fluids move a little bit differently, and we're going to spend a hot minute on this slide, so prepare. Okay, I'm going to write a lot of things on this slide. But what we're talking about right now is this method of transport called bulk flow. In bulk flow, there's two main pressures that we care about. The first one is called hydrostatic pressure. Hydrostatic pressure, or HP, and then like a little subscript C, okay? So hydrostatic pressure, hydro, think like water or fluid. That's the pressure that we push on those vessel walls. So I kind of think of it like blood pressure, like how hard that blood is pushing on the walls of the vessel. So the pressure at which fluid is pushed against vessel walls. And what hydrostatic pressure does is because it pushes on the walls of that vessel, hydrostatic pressure promotes this word called filtration. So hydrostatic pressure promotes filtration. Filtration is when fluid is leaving the vessel. So fluid is leaving the vessel. And this, of course, is in capillary, so it would be leaving the capillary. So I'll highlight that new word, filtration. I'll highlight it in red. Filtration, that's a new word. But the next pressure that aids in bulk flow is called colloid osmotic pressure, or osmotic pressure. And the abbreviation here is OP with like a little subscript C. So osmotic pressure is kind of like, I think of it like sucking. It's the pressure pulling stuff in, and the stuff that we're pulling in is fluid, of course. So it's a sucking force. Or a sucking pressure, rather, is a better word for it. Sucking pressure. That draws fluid towards the area, of higher solute concentration. So it's the sucking pressure that draws fluid towards the area of higher solute concentration. So wherever the solutes are higher, that's where the fluid goes, okay? And I think of colloid osmotic pressure, I want to think of that word osmosis. We have to think back to that word osmosis, the diffusion of water, but it's really that we want to go to the area where water is lower. Okay, so let me highlight colloid osmotic pressure also. Okay, so how I'm going to show you about this is first we need to draw a blood vessel, and well, it's capillary, okay? So here's my capillary. I'll draw like this and like that. Okay, so there's my capillary. I'll label some stuff here. I'll do all my labeling in black, and then we'll do all our annotations in something else. Okay, so this is the lumen. So capillary lumen. And then on both sides, so here and here, we have the same thing going on. I'm just going to draw on one side just for easier. So here is my interstitial space. Or ISS. And if blood flow goes this way, if blood flow is going to the right, then we could say that this is the arteriolar end. Because remember, we go from our arteries to arterioles to capillaries and then to venules and veins. So this would be the side of the arteriole. And then that would make the other side the venule, the venular end. Okay. We know that the pressure on the arteriolar side is higher than the pressure on the venular side because the pressure in arteries is bigger than the pressure in veins. This is something that we know. And so what happens here is that we have also some stuff inside of the blood. We have cells and stuff, but we also have little proteins. So I'm gonna draw these little, I'll draw them in black. Little proteins. These little black bubbles that I'm drawing are plasma proteins. And the plasma proteins, they're just swimming around in the blood, so they're kind of consistent from the arteriolar end to the venular end. So I'll label these. These are plasma proteins. Plasma proteins. And plasma proteins are made by the liver, and when we get into the blood, we'll talk more about them. So the other thing that I want you to know is that when I'm drawing, so when I draw an arrow, we're going to draw arrows, and I want you to remember that the size of the arrow is equal to the relative amount of filtration, or a new word is reabsorption. Or reabsorption. That's so filtration is when fluid is leaving the vessel, and then reabsorption is going the opposite direction. Okay, so I think we're ready to start talking about things. Okay, so on the arteriolar end, what's happening with our hydrostatic pressure is that hydrostatic pressure is bigger. It's blood pressure. It's the pressure of fluid pushing on these vessel walls. So that hydrostatic pressure is pretty big. So our HPC, our hydrostatic pressure of the capillary, is pretty large. It's bigger than our osmotic pressure. So our OPC. In fact, our OPC, our colloid osmotic pressure of the capillary, is pretty consistent throughout. So OPC, around, I'm drawing that like trying to, around the same size. So those are the same. But what happens from the arteriolar end to the venular end of a capillary bed is that we really decrease our blood pressure, our hydrostatic pressure. So the pressure at the venular end pushing on that vessel walls is just weaker, because we've passed through this whole meandering thing of capillaries. It takes a minute. So what happens is at the venular end, that HPC is actually smaller. So the osmotic pressure is bigger on the venular end. Notice like the size that I'm drawing. So when hydrostatic pressure is bigger, as it is on the arteriolar end of the capillary, hydrostatic pressure is the pressure that pushes on these vessel walls. And so if hydrostatic pressure is pushing on those vessel walls and capillaries are permeable, we're going to start pushing fluids out. And pushing fluids out is the process called filtration. So I'm going to draw my big arrow like this, like that. And then I'm going to get my little eraser and erase all the stuff inside. So that arrow pushing stuff out is called filtration. So stuff starts to leave the vessel. Similarly, same business on the bottom. So still filtration. So blood is going to leave that vessel at the arteriolar end. Stuff gets pushed out. What happens on the venular end is the flip side. Because osmotic pressure is bigger, we're going to pull stuff in. And why we pull stuff in is osmosis. Osmosis happens when we have a different concentration of water across a thing, okay? So in the interstitial space, there's lots and lots of water, not so many proteins and solutes and things. So what happens is solutes draw water. We learned that in Bio 430. So anytime there's lots of solutes, like plasma proteins, then that's going to pull the water inside. And so that's what happens. But this arrow is smaller. This process is called reabsorption. Reabsorption, same thing on the other side. Absorption. So that's pulling fluid in. And remember that in this interstitial space, what's here is not a bunch of proteins and stuff, it's a bunch of cells. This is the tissue. These are our tissue cells. So this is really, really important because we're going to deliver things via filtration. Deliver it to those cells through that space. And we're also going to get rid of stuff that we don't want, and we're going to put it back into the blood. So on average, if we were to assign these numbers, what would happen is on the arteriolar end, we would actually filter about 20 liters of fluid on average. We would reabsorb 17 liters of fluid on average. So 20 liters and 17 liters, that's not the same number. So we have some fluid that remains in the tissue, in the interstitial space. And that fluid gets removed by this other thing that I haven't told you about yet. This other thing that's there, which is called a lymph vessel. So I'm just kind of draw it like a little finger sticking out. This is called a lymph vessel. Lymph vessels, they pick up excess fluid. So lymph vessels carry away excess fluid. And we'd carry away that extra then 3 liters of fluid away. And that's because the filtration is exceeding the reabsorption. And so that lymph vessel is just kind of around to be like, hey, I'll take your extra fluid whenever you got it. Okay, I'm going to zoom out on this side so we can get the whole thing. Capillary exchange of fluids happens ever so slightly differently than respiratory gases, the nutrients, the metabolic waste, which happen because of diffusion. But exchange of fluids happens with bulk flow. Your textbook has a really great figure to illustrate this. It does have some math, so you don't need to look at the math because we don't really care about the math so much for class. But we will experiment and do some things with this in the lab. All right, I hope you enjoyed this one.